Now it's my pleasure to introduce our guest speaker. Douglas Vaughan is the chair of the Department of Medicine and Irving S. Cutler Professor of Medicine at Northwestern's Feinberg School of Medicine. He also serves as physician-in-chief at Northwestern Memorial Hospital. Today, Dr. Vaughan will discuss the fascinating research on aging. He'll explore whether some parts of the aging process are preventable. I'm most interested in that. How the aging process unfolds in humans, and how researchers are using targeted approaches to preserve people's quality of life as they or we age. Dr. Vaughan's presentation is entitled Aging, Inevitable or Preventable. Please join me in welcoming Douglas Vaughan.
Thank you very much, John. Good morning, everyone. Why is anybody interested in aging? If you're not interested in it, you've got a problem. I'm not going to talk about any magic coffee beans this morning, like you might hear on TV, or drugs from jellyfish. But I'm going to talk about a process that impacts on all of us, and it's the aging process itself is unfolding now. And when I talk to our medical students and my house staff, I say this is the most exciting time for anyone to be in medicine, because over the next 20, 30 years, we're going to see many, many cancers become not just treatable, but curable. It's happening every day now. And we're going to get our hands around the aging process in a real way, and we're going to be able to slow that process in many human beings. So aging is important in medicine. Let me tell you a couple things that sort of set the optics for you and why we're interested in it in the Department of Medicine. First, I'm a cardiologist. What's the most important risk factor for heart disease? You know? It's age. It's not smoking. It's not cholesterol. It's age. What's the most important risk factor for cancer? Age. What's the most important risk factor for neurodegenerative disease? Age. So there's a common denominator here. You guys are all smart. You went to Northwestern. You can figure that out, right? And the reality is it's not how many times you spin around the sun. There's a chronological age that you can quantitate and measure from the day that you're born, but there are differences in biological age. And we see it every day in the wards at Northwestern and every other hospital in the country. We see people with diseases that make them age very rapidly. We see people that come in that are old and look much younger. And so there's clearly something going on that allows you to pivot or alter the trajectory of your individual aging process. And that's what I want to talk about with you this morning. Okay? Have you seen this billboard around town? First person to live 150 years alive today. I was looking at John. I was thinking he might be that guy, but he looks pretty good. It's a pretty interesting comment, isn't it? Aubrey de Grey is an internationally known researcher in the field of aging. He's iconoclastic. He's got a great TED Talk if you want to look it up. He says we have a 50-50 chance of bringing aging under what I call a decisive level of medical control in the next 25 years. I think, he may be wrong, but he may be right, actually. This is the holder in the Guinness Book of World Records for the longest living human. This is Jean Clement. She did die, though, like every human does. But she made it to 122 years. 122, plus a few days. Pretty remarkable, and she was quite vigorous up until the time she died, and many of the photographs that you can find of her online show her smoking a cigarette, actually. There are a lot of people that are getting interested in aging, and it's important when Google gets involved, right? Can Google solve it? Well, they put $500 million into a new company called Calico. It's one of their moonshot agenda things, and their Calico is all about understanding aging and developing therapeutic targets and products that might slow the aging process in human beings. I think this comment by one of their leaders is, here's to hoping we see some results in a short decade or two, before it's too late for us. That's the problem. It's inevitable that you're going to age, and even if you try to slow it down, you better get involved in the process pretty early. So in the world of medicine, we see people with accelerated aging all the time. There are very rare genetic causes of aging, like progeria, and these children are about 1 in 6 million in the world. There may be 200 or 250 of them in the world that have this, but they age right in front of your eyes. They rarely live beyond 12 or 13 years, but if you look at them, they look like they age very, very rapidly. They've got a very distinct and rare genetic mutation. There are other more common mutations in the telomerase complex. This is the complex that maintains the integrity of the length of your chromosomes. This has just really been discovered in the last 20 years. People with telomerase mutations can age rapidly too, but not like people with progeria. They'll have premature gray hair. I don't want to scare anybody in the room today. Oh, and they also develop bone marrow failure, pulmonary fibrosis, often liver failure too, sort of for unknown reasons. But that's a more common cause of something that looks like rapid aging. And then we see aging accelerated in many common clinical conditions, actually. People with chronic kidney disease are probably the most remarkable example. If you look at someone on if you look at a 25-year-old on dialysis, they have the same cardiovascular risk as a 75-year-old individual. So they rapidly accelerate their aging process of their cardiac system and their vascular tree. People with diabetes and obesity can age more rapidly. People that smoke cigarettes age more rapidly, although the woman I showed you probably didn't. She was refractory to that. People with chronic HIV infections look like they age more rapidly, even if you control their viral load. So we see it every day. We see it every day in the hospital. The students just presented a case to me this week of a 37-year-old woman with end-stage liver disease, and she was terribly sick. But I asked them to describe, how does she look? And they said she looked 90. She's got a chronic aging syndrome based on her underlying diseases. Well, it turns out the reality is we can start to measure biological aging. There are ways that we can actually estimate it and quantitate it in a meaningful sort of way. So there's a real molecular and biochemical fingerprint of age that we can all look at. It's pretty simple. And having that fingerprint, having that knowledge, gives us the possibility to be able to measure it and actually see if we can impact upon that process. Make sense? So the most widely accepted measurement of biological age or aging is your telomere length. So what's illustrated on this slide is the fact that as your cells divide, every time cells in your body divide, the chromosomes get a little bit shorter because the polymerase complex can't get out to the ends. So in all of our adult cells, the chromosomes gradually get slower. And the length of your telomere will decrease as you age. People with that telomerase complex mutation have very short telomeres to start with, and they age very rapidly. So it's a very unique and important molecular marker of biological age. Now there's all kinds of controversy about this. Can you really change the rate of your slope over time, or is it set by the time you're a teenager and you're just on your descent slope for the rest of your life? It seems like likely both things are true, but there's still work to be done. But clearly it's a great measurable and quantitative marker of your biological age. So if you look across the ages, you can see from neonates to middle-aged people to older people, you'll find differences in the length of their telomeres. The best example about the relationship between telomere and age comes from our best friends, canines. What's the old saw? A dog's ear, 6, 7 human years? Well, that's based on telomere biology. You may not know that, but it turns out that the lifespan of a dog is largely predicated on their average telomere length. So larger breeds, like Great Danes. Anybody have a Great Dane in the audience? They tend to have pretty short telomeres, and they have a pretty short lifespan. Other dogs have longer or more variable lengths in their telomeres, like beagles and poodles, and they tend to live much longer. But clearly there's a relationship between telomere length and lifespan in canines. Along with the idea that you can have a molecular measurement of biological age, there are also biochemical measures of a process that we call senescence. Now, it turns out senescence is critical in all of us. As we age, one of the things that we lose is our regenerative and our reparative potential. It's true in our vascular tree, it's true in our lungs, it's true in all the tissues in our body. And it turns out there are markers of cellular senescence that you can identify. Cellular senescence is a defined process. There are distinct molecular pathways that drive cells to become senescent. And what is a senescent cell? A senescent cell has lost its ability to replicate over time. It's lost its ability to divide. And it's thought that senescence is one of the main drivers of aging in animal species. There are lots of things that can initiate the senescence pathway in animals. Things like inflammation, ionizing radiation, oxidation, all these kinds of things that we're exposed to in real life as we go through our life. And all of those can impact upon these pathways that impact upon cellular senescence. One of the key molecular pathways involves P53. You don't need to be a molecular biologist to know about P53. Mutations in P53 are associated with many different kinds of cancer. So if you lose this senescent pathway, you get the other side of the coin, which is hyperproliferation or cancer. It sort of makes sense. And actually, most of the people, many of the scientists that work in the field of senescence came at it from a perspective of working on cancer. They're trying to find a way to flip cells into a senescent paradigm instead of a proliferative paradigm. Now, it turns out senescent cells express or synthesize a variety of different proteins that you can measure. It's a distinguishing fingerprint of senescence as well. And there are a lot of different proteins that have been identified, things like TGF-beta and an insulin-like growth factor binding protein and interleukin-1, which is an inflammatory protein, and a protein called plasminogen activator inhibitor type 1, or PI1 for short. So when cells get pushed down this senescent pathway, you can identify them in tissue culture or in animal models by virtue of the fact that they make proteins like these. So another measurable, quantifiable aspect of the aging process or the senescence process is relegated or is identified by these particular proteins. Now, I'm a cardiologist. Before I came to Northwestern 8 years ago, I was at Vanderbilt, and I worked on vascular biology and cardiovascular disease for years. When I moved to Chicago, I really kind of sat down for a while and thought about my science and my career, and I wanted to really work on something important. Not that what I'd been working on for the 20 years before I got here was trivial, but it was incremental. And I really thought, and I said, you know what? You're really working on aging. You don't call it that, but that's what you're working on. And so when we moved up here, we changed and reoriented the laboratory, and we said, you know what? We're really going to focus on human aging through the window of the role that PI1 might play. So PI1 is one of those markers of senescence. It's the protein and the gene that I've been working on for 30 years. PI1 is the natural inhibitor of your clot-dissolving system in your body. You can imagine why a cardiologist might be kind of interested in that. It's the natural inhibitor of your own tissue plasminogen activator, the clot-dissolving drug that's commonly used in clinical practice We used to use it to treat heart attacks. We now use it to treat strokes. It allows you to dissolve a clot inside your vascular bed. Pi1 is your natural inhibitor of that system, but it turns out Pi1 is a marker of senescence. So we started to try to link those two features up, and we started asking the question, is Pi1 just a marker of senescence, or does it contribute to senescence? Other people have been interested in this as well. I'm not going to get in great detail here, but what I want to show you here is how really this was a pivotal publication that sort of triggered our thinking around Pi1 and aging and senescence. This was published several years ago. But what Kortlever and colleagues here are illustrating is the fact that when cells are grown in culture, they eventually become senescent. They stop to replicate. It's called the Hayflick phenomenon. And if you take normal cells, you can see they stop dividing, they stop proliferating over time. It's a very reproducible phenomenon. Now, if you take cells and you take p53 out of them, that molecular component that drives the senescent pathway, if you take p53 out, those cells escape replicative senescence. They're malignant, if you will. It turns out, if you take Pi1 out of those cells as well, they start more slowly, but actually they can escape replicative senescence as well. So this suggested that Pi1 was not just a marker of senescence, it actually contributed to senescence, at least at a cellular level. And this was an aha moment for us with regard to our own research and where we were headed. So let me tell you a little story about where we've gone with this over the last few years and where we're headed. So it turns out there are all kinds of murine or mouse models of accelerated aging that are available now. Mice that have something that looks like progeria. There are all kinds of mice with various mutations that look like they age pretty rapidly, and that aging might be manifested by cardiovascular changes or changes in the lung or changes in their behavior or other kinds of phenotypic changes. About 20 years ago now, a group in Japan were attempting to make a transgenic animal. It's not important really what it was, but they popped a new gene into a mouse line, and when they looked at the mice, it was nothing like they expected. The mice stopped growing after 3 weeks. This is one of their original. This is what the mice look like compared to a wild type or unaffected animal. And it turned out these mice, they called them Clotho because they ended up, and I'll describe it to you in a moment. And who was Clotho? Who remembers anything from college? Clotho was one of the Greek goddesses that controlled the thread of life or your lifespan. Clotho was actually the spinner of the thread of life, and she worked together with two other goddesses to determine the lifespan of mortals. So the Japanese investigators that engineered this new mouse line called it Clotho because it looked like it was impacting upon the thread of life or the lifespan. And what do they look like? Well, they have muscle atrophy, they have skin atrophy, they have neurodegeneration, they have hearing loss, they have a short lifespan. They live about 10 weeks. A normal mouse, if you don't bother them, will live about 3 years. So very shortened lifespan. They're sterile. They have emphysema. They have renal sclerosis and calcification in their vasculature, and they get osteoporosis. Sounds like aging to me. And one of the interesting things about Clotho is they show an age-dependent increase in plasma and tissue levels of Pi1, just like they have senescence cultivated or smoldering in them and driving part of this phenotype. So that made me have a big idea. This is a picture of me when I had my big idea. So we asked the question. So Clotho animals age rapidly. They have a high level of Pi1. We think that Pi1 contributes to senescence and aging. What would happen if we take Pi1 out of Clotho animals? Pretty simple. All that involves is some breeding of clotho-deficient animals with Pi1 deficient animals, which we've had in my laboratory now for 15 years. And we just wanted to see what happened. Well, right away we saw some pretty interesting things happen. Here's a clotho-deficient animal from our laboratory in Chicago. Here's an animal that's partially deficient in Pi1. They're missing one of the Pi1 genes. You can see it's grown to a much more normal size compared to its littermate counterpart. Here's an animal that's missing both copies of Pi1, and it's not much different than the animal that's heterozygous. The animals that miss either one or both copies of Pi1 gain more weight. Over time, they're more active. That's one of the things that you can measure that changes as you age, whether it's a human being or a mouse. Your activity levels go down. So it looks like we rescued the clotho animals in a significant way by taking Pi1 out of the equation. And then we looked at the tissues that age very rapidly, and we're particularly interested in a few, but for example, in the lungs, this is normal lung. This is lung from a clotho-deficient animal. You can see it's got these bigger air spaces compared to this. This is emphysema. And if you take Pi1 out of the equation, either partially or completely, we largely protect those lungs from those aging-like changes. And we even preserve the ability of the lungs to exchange gas. Here we're measuring the oxygen content of the blood, and we rescue that by taking Pi1 out of these animals. So that's pretty good. We also looked at molecular markers of senescence and aging in these animals. Here's an example of that. This is kidney tissue, and here we're staining for a molecular marker of senescence called P16. So P16 is expressed in cells as they become senescent. There was a fascinating paper in Nature just 2 months ago by a group at the Mayo Clinic. They selectively killed cells in the body of an older mouse that were expressing P16, and they made those mice actually look younger. Kind of reversed some of the aging changes in their cardiovascular system. So P16 is a relevant sort of marker of aging and senescence. But when we took Pi1 out of the mice, we saw a marked reduction in the expression of P16. That's the dark coloring here. And if we measured it quantitatively, we can see wild-type animals don't express very much. P16 goes up a lot in the kidney of clotho-deficient animals. If we take Pi1 out of the equation, we largely shut down that molecular signal of senescence. And here's the most important part, and this is the answer to the question I hope you already had in your mind. Well, okay, you preserve the lung structure and function, you reduce the molecular changes of aging in tissues. Oh, well, what's it do to their lifespan? Well, actually, we take Pi1 out of the equation in clotho-deficient animals. So here's the survival curve of clotho animals. See, they're all dead by a little over 100 days. Their median lifespan is about 8 weeks. But if we take Pi1 out of the equation, either partially or completely, we can quadruple their lifespan. Not trivial. That p-value is 0.00001. Okay, Dr. Vaughan, interesting story. Mice are cute. What's this got to do? What's this got to do with human beings? Does this actually have anything to do with human beings? How would you ask that question? Anybody got any ideas? I can't go knock Pi1 out of John, even though he might like for me to. We're not going to manipulate embryos, take Pi1 out and see what happens. You'd have to wait 100 years. What are you going to do? Well, nature does some interesting experiments. Turns out in eastern Indiana, over here in Bern, Indiana, there is a group of old order Amish who originally settled that region. They came from Bern, Switzerland in the middle of the 1800s. And they called where they settled in Indiana Bern. I'm pretty sure it looks just like Bern, Switzerland. No, it doesn't. We've been there. It's a 3-hour drive from Chicago. Turns out in 1992, a young girl was identified that was part of that kindred that had a weird bleeding disorder. She didn't bleed spontaneously. She wasn't a hemophiliac. But after she was injured or after she had minor surgery, she would form a clot just fine, or she'd form a scab on the surface of her skin. But a day or two later, she would start oozing and she would start bleeding. And so a couple of hematologists in Indiana saw her, and they worked her up, and they found out that she had no Pi1. No Pi1. Her clot dissolving system, which I talked about originally, her clot dissolving system worked too well. It wasn't inhibited by Pi1. So they went and examined the whole kindred, and they found that there were lots of individuals that either had partial or complete Pi1 deficiency in this kindred. 12 individuals have been identified so far that are completely null for Pi1. There are over 110 individuals that are heterozygous, or carry one mutant allele, if you will, for the Pi1 mutation. As far as we know, this is the only group like this in the world, in the solar system, in the galaxy, as far as I know. But there they are. They're 3 hours away from Chicago. We can trace back the pedigree of the kindred. The founder with the mutation actually came from Switzerland, and he had 15 children, and he had 148 grandchildren, and I can't even tell you how many great-grandchildren he had. The first proband, the little girl that was identified with the bleeding disorder, was 5 generations removed from that founder, and her parents were remotely related, but they both carried one copy of the mutant gene. So that's, and there are other aspects of their culture and their life that make this possible. They're socially isolated from the rest of Indiana. Not that that's all bad. They're socially isolated. They're almost chronologically isolated from the rest of us. They live an 18th century lifestyle. No combustion engines, no electricity. They get around by horse and buggy. And they don't move. They don't marry people outside of their community. So it's like they're on an isolated island in the Pacific somewhere, but they happen to be 3 hours from Chicago. So here's how how do we take our mice data and test it in human beings? Well, nature's done that experiment for us in Bern, Indiana. So I got a grant a couple years ago to go study them. And the National Institutes of Health agreed with us that we thought it'd be interesting to go test the hypothesis that these people might be protected from aging. In particular, we wanted to look at individuals that carry one copy of the mutant gene as opposed to 2 copies. The girl that I talked about that was originally described as carrying the double mutation, she's just 34, 35 years old now. And there are only just a few of those homozygotes, as you will. But there are a lot of heterozygotes. And they're kind of the interesting group, if you ask me, because they speak to what might be possible in the real world if we were to target Pi1 in a different kind of way. And I'll talk about that in a few minutes. So last May, I took my research team and we met with the hematologists in Indiana, and we set up shop in a recreation center outside of Bern, and 177 of members of this Amish kindred came and visited us over a 2-day period. And we took over all kinds of equipment from Chicago, echocardiographic machines, blood pressure machines, people to draw blood, people to spin blood, people to isolate DNA, do all kinds of things. And we spent 2 intensive days putting them all through the paces after we received informed consent from them. And we measured a variety of things to try to quantify their biological age, if you will. These are some of the things that we did. We gave them a questionnaire for their fitness age. We measured their height, weight, their waist circumference. You don't see many obese people in the Amish population. They work pretty hard. We did things like measured how stiff their blood vessels were. That you can do that non-invasively. We did echocardiograms on everybody. We looked at their heart structure and function. We measured the thickness of their carotids. We measured their pulmonary function. We even took pictures of their retina. They won't allow you to they don't like to have their faces photographed. That's kind of verboten in the community, but they would allow a photograph of their retina. And we collected urine and other things. The age distribution of the people that we saw is shown for you here. We really looked at adults. We saw several individuals that were greater than 70 years of age, but it was a pretty nice distribution of age overall. And what we found, what came to visit us over those 2 days included 7 of the homozygous individuals. In fact, I got to meet that young woman who's now older now, who was the original proband with the deletion. We had 43 individuals that were heterozygous for the mutation and 127 of their cousins who don't carry the mutation at all. And you can see that they're, particularly when you compare the heterozygotes to the unaffected individuals, you can see that here's their percentage of women. Their systolic blood pressure tends to be a little bit lower. Their diastolic blood pressure is the same. The incidence of hypertension was lower in the heterozygotes. And interestingly, the incidence, they have no diabetes in the heterozygotes, none. And that's something we're chasing right now, but that's going to be another part of the story. Our primary endpoint, the one we designated before we went to Indiana, was we hypothesize that the carriers of one mutant allele will have longer telomeres than their cousins. All right, that means that their biological age would be younger than their cousins. It turns out it's true. Here's the plot of telomere length over time as a function of age. You can see that the lines diverge, and this is highly statistically significant. So actually their telomeric length, as a reflection of their biological age, is significantly younger. We did a combined endpoint looking at all those cardiovascular parameters I talked about, carotid IMT, pulse, or vascular stiffness, echocardiographic things. When we put them all together, we find that the individuals that carry the mutant allele have a more juvenile cardiovascular system compared to their cousins, and controlling for age, gender, and family relatedness. So this is independent of all those things. We found a couple other things that are really, really critically important when you think about aging. We found that the heterozygotes had a nearly 30% reduction in serum insulin levels. That's highly statistically significant. And most importantly, we're able to dig into the pedigree and figure out who's who, what's what, who had the gene, who didn't, and we've been able to go far enough in the pedigree to know that the carriers of the mutant allele live about 10% longer. And that's highly statistically significant too. So we just finished writing this paper 2 weeks ago. It's out being reviewed at a big important journal right now. You're hearing about this data. You're the first people I've really ever talked to about this. But this represents the first gene mutation in human beings that protects against biological aging. I don't think that's trivial. I think it's a really big deal, and it starts to give us an opportunity to think about how we'll impact upon aging in human beings in the future. So we see that they're younger. We see that their aging is delayed at a number of different levels. At the molecular level, we see that they have longer telomeres. At the tissue level, we see that their cardiovascular system looks younger. It's not as stiff. It works better. At the systemic level, they have lower insulin levels. And many people that work on aging, whether it's in worms or flies or mice, think insulin and the insulin axis are critical in driving the aging process. So a lower insulin level is a remarkable finding in this population. And the fact that at an organism level, they live longer is remarkable as well. So, okay, I've talked about mice, I've talked about aging, I've talked about this very, very unique and remarkable population in eastern Indiana. Now what do we do? What do we do? How would we ever take this? How would we ever do anything in human beings? How would we ever test this? Together with Toshio Miyata, he's my partner. He's at Tohoku University in northern Japan. They were up around the epicenter of their huge earthquake a few years ago. He's also adjunct faculty now with us at Northwestern. Toshio's a nephrologist, and he was interested in Pi-1 for some different reasons, but he's got a drug discovery unit at his university. And several years ago, they embarked on an adventure to develop some drugs that block Pi-1. Small molecules that specifically target the protein that can be taken orally that inhibit Pi-1. And he was developing them for a variety of different reasons, had nothing to do with aging. But he contacted me a few years ago because we know a little bit about Pi-1, and we started a partnership that we've successfully built and established over the last several years. So this is the Pi-1 protein in a two-dimensional rendition. And what Toshio and his colleagues did is they used a virtual screening strategy. So they know what Pi-1 looks like in 3 dimensions. It's been crystallized. And they using computers and in silico, they tried to find molecules that could fit into this groove of the protein. And some of these molecules are illustrated for you here, thinking that that might be a successful way to inhibit the protein. And they screened over 3 million molecules in silico, and they narrowed it down to about 80 different things that looked like they might fit there, and then they synthesized those or bought them and successively tested them for their ability to inhibit Pi-1, and then they revised them, chemically modified them, reiterated, retested, and gradually they developed some drugs that actually looked like they may work. And we tested them in a model that we've had in our laboratory for several years. We made a model about, while I was still at Vanderbilt, a mouse model that expresses an extra amount of Pi-1. And it's not mouse Pi-1, it's human Pi-1. We made this mouse almost 15 years ago thinking someday somebody's going to come up with a molecule to block Pi-1. And they're going to be interested in whether or not it can inhibit human Pi-1, not mouse Pi-1. So we made a reagent, if you will. And we've tested several potential drugs from a variety of different drug companies over the last several years. Nothing ever worked. Nothing ever did anything in our mice. But then Toshio contacted me about 6 years ago and asked me if we would test his drugs in our mice. Now, our mice have some very characteristic abnormalities that they've had durably the whole time we've had them, whether in Nashville or in Chicago. They're partially bald. You can pick them out of the cage. It's easy to tell them from their counterparts that don't have that transgene, and they've always been that way. And they develop amyloidosis. They deposit protein in their liver and some in their brain too that is sort of suggestive of an aging process actually. And one of the things that these mice do, quite surprisingly, is they'll have a heart attack. They will spontaneously clot their coronary arteries. So in the history of the world, I challenge you to find me a mouse that's ever had a heart attack. You know, it doesn't happen. You know, what kills mice? Traps, cats, investigators like me. But they don't have heart attacks. But these mice that overexpress too much human Pi-1 actually will clot their coronary arteries, and they don't even have atherosclerosis. So in a lot of ways, and in fact, we said this from the first day we saw these mice in the laboratory in Nashville. We said, they kind of look old. They're bald. They have heart attacks. They got this problem with their liver, you know. But we didn't put 2 and 2 together. It took some time. So 6 years ago now, Toshio sent me some of the drug, and we just simply put it in the chow of the animals. We just fed it to them in their diet. And here are a bunch of animals that have not been fed the drug, and here's the animals that have been fed the drug. I said, I won't say exactly what I said. They grew the hair. They grew hair. I said, I'll be darned. His drug actually works. It inhibits human PI1. That's what blocks the hair growth in these animals. So then we started testing it in experimental and genetic models of aging, just like you'd expect. We can make animals develop arteriosclerosis. So this is an aorta from a mouse that's had its ability to generate nitric oxide inhibited for several weeks. And this is an aorta from a normal animal. You see all this blue staining? That's arteriosclerosis. That makes an artery stiff over time. That's what happens in all of us as we age. But if we give the animals the drug that causes arteriosclerosis, and we give it with the inhibitor of PI1, you can see that vessel is pristine. It looks fantastic. And we can even measure a reduction or a blunting in the change in blood pressure over time. Let me tell you, if you live long enough, you're going to have high blood pressure. All of us, our pipes get stiffer as we age. And this is largely due to this arteriosclerotic kind of phenomenon. These mice also develop emphysema. You can see these big air spaces. And here's what happens if we give the drug with the other one that causes the emphysema. We see we largely protect the animals. Or if we give L-name to PI1 deficient animals, they're largely protected too. So the drug protects against a couple manifestations of aging that affect all mammals. Then we went back to our clotho animals, those rapidly aging animals that showed all those kind of phenotypic abnormalities I talked about a little bit ago. And we simply put the drug in their diet. And what happened there? Whoa, quadrupled their lifespan. And Toshio and his colleagues in Japan have recently given the drug to a different model of accelerated aging in mice, mice called sirtuin 6 knockouts, and they quintupled their lifespan. So where do we go from here? The National Institute of Aging has a program where they'll test drugs or dietary supplements to see if they can prolong the lifespan of a normal mouse. And that's really kind of key. You want to know, not does it affect an experimental model of disease or a genetically modified animal, what's it do in a regular healthy mammal? Does it make them live longer? And we suggested that they try the drug that we're helping develop. Actually, they selected it. So it's under testing right now. And they tested at 3 different locations around the country in large numbers of outbred, wild-type healthy mice. And they looked to see how many of them live beyond 3 years of age. They're about a year and a half into studying the drug. And they tell me that we may know something by December of this year. But the important question is, what's it do in a healthy, normal mammal compared to a sick mammal or a genetically modified mammal? And if you read the paper, if you watch the news, you can see the idea of developing therapies that impact upon aging is starting to bubble. It's not boiling yet, but it's getting pretty interesting. You'll hear about things like sirtuin activators, and that includes resveratrol, which you can find in red wine and other things. A drug called rapamycin has been tested in that National Institute of Aging program in mice, and it actually prolongs lifespan in healthy wild-type mice. But rapamycin is an antibiotic that comes from a fungus. It's got some side effects. I don't think any of us are going to want to jump on rapamycin anytime soon, even though it's an FDA-approved drug. IGF pathway modulators are thought to might be very interesting down the road. I think that's what Calico and other drug companies might be working on. Caloric restriction is the big thing that it's known to prolong lifespan on all kinds of things, from yeast to mice. Simple caloric restriction will prolong lifespan. Studies are not definitive in primates so far. Primates tend to get a little aggressive if you take away their calories. No surprise. So we'll have to see what happens. Yeah. Drugs that block the senescence messaging secretome or proteins that are generated by senescent cells like PI1, I think are going to be interesting. There might be other targets that come out of that fingerprint of senescence. And drugs that reactivate telomerase in all of us or in our somatic cells might be interesting as well. So I want to wrap up. I think, how to live forever. I don't know if that's on anybody's radar screen. Going to need a lot of candles. But I think, it's a pretty exciting time in medicine. It's a very, very exciting time in biology. I don't think there's any doubt about it that the next 20, 25 years are going to see us have the ability to slow down the aging process in people. But I think Aubrey de Grey hits the nail right on the head. It's not about keeping people alive in a bad state of health. That's not what this is about. It's about preventing people from getting sick just as a result of aging. And I think that makes more sense. I got to recognize many of my laboratory group that have worked on this, including Sadia Khan, who's a Northwestern graduate from undergraduate and medical school, and she's my fellow on this work. This is my partner, Toshio Miyata, and these are the hematologists in Indiana that we are indebted to because they're the ones that discovered the PI1 deficiency in the first place, and we would never have known the address of any of those Amish to invite them to meet with us if it hadn't been for them. So thanks a lot for your attention. What part of the human body produces PI1? In general, it's the liver. It's also made in visceral fat. So your body mass index is the best predictor of your plasma PI1 levels. It's got a relationship like that. The higher your BMI, the higher your PI1 levels. So that sort of makes sense. You think about caloric restriction, maybe have a beneficial effect. One of the things caloric restriction is going to do in mammals, in mice, and in humans, I'll guarantee, is lower your PI1 levels. So visceral fat's the most important predictor of your plasma PI1 level. Yeah.
I understand you to say that PI1 deficiency results in a kind of hemophilia.
Yeah. He's asking about does PI1 deficiency result in a kind of hemophilia? Yes. If you have 2 mutant copies of the gene. People that only have 1 copy, no, they've been followed now for 20 years, they do not bleed. Okay, and that gives us hope that you could target that and just lower it. I don't think nature wants you to have 0, but I think you could have a normal level or a lower level and be very, very safe. And so that gives us confidence going forward. But a good question. Yeah.
Besides PI1, is there any truth to taking like antioxidant supplements and things of that nature? I mean, I know we should all have a good diet, exercise properly. It's different for everybody in this room. So, but just so that we're all here again, like about 50 years,
Yeah.
all of us, is there any truth to that? Taking supplements?
Asking about supplements, particularly antioxidant supplements, and do those have any impact upon the issues that we're talking about today? I don't know. I'm not a big fan, actually. I think, at least in the cardiovascular world, the administration of high-dose antioxidants like vitamin E have not been effective in lowering rates of cardiovascular disease. So I don't know. I mean, certainly oxidation is a player, and it contributes to and can push cells down a senescent pathway. I don't know that we have the right kind of antioxidants on the market now. And I don't think you can go buy them off the shelf of your pharmacy or your anything else. Yeah.
Does physical activity have any effect on the levels of PI1 in a person's body?
Question is, does physical activity have any effect on PI1 levels? It certainly does, especially if you do that and you lose weight. So I think that's the relationship there in general. I think exercise is one of the ways you can kind of keep yourself young too. Yeah.
Do you notice any side effects or negative repercussions from
The best example are the heterozygotes in the Amish community, and the heterozygotes look perfectly fine. If they had not generated some homozygotes, you'd never know that they had this gene mutation ever, because they look. Actually, they're healthier than normal, and they don't have a problem, any problem that we can see so far. Now, Toshio and his colleagues have taken the drug into human beings already in Japan. They've given it to 100 healthy volunteers in phase 1 studies, and everything's okay. So their Japanese regulatory agency has given them permission to move into phase 2, which is pretty interesting. I mean, they're not doing an aging study or anything like that, but the drug looks like it's, safe, at least in short-term administration so far. Yeah.
Has there been any research looking in parallel, I don't know, when you were talking about just general physiological health, how about mental health?
Yeah.
Disability in that regard?
The question is, are there any, what kind of studies have been done related to cognitive function, mental health, et cetera? We didn't do any kind of cognitive testing in the Amish. We want to get a grant, go back and do some other things that we couldn't do on the 2 days that we were there before. We want to do things like measure their hearing, do some cognitive testing. They speak English, but with each other, they speak a low German dialect that's completely unintelligible to me, other than V. Gates. Large population studies have been done using genetic analyses, looking for polymorphisms that might protect against aging or cognitive dysfunction over time. The one thing that's popped out in large population studies are mutations or polymorphisms in ApoE, which is associated with Alzheimer's disease. So some mutations in that appear to be protective. In the large population studies, this Pi1 thing has not popped out because it doesn't exist in large populations. We've looked in large databases with 120,000 human beings fully sequenced, and this mutation is unique to the Amish. And there's no, as far as we know, no other human beings have it. Yeah.
So there are like what's termed as blue zones where
Mhm.
certain areas in the world where you have people that live longer.
Yeah.
Has there been any investigation as to what is in existence and not in existence from a Pi1 level?
Yeah. The question is, so you may have read or heard about these blue zones in the world, localized geographic areas where people tend to live longer. And the question, has Pi1 been looked at in any of those populations? Not that I know of. Those are interesting. I think that there's probably some reality to that. It's multifactorial. It's probably cultural. It's probably diet. It's probably genetic too. I mean, if you think about it though, the most complex human trait is your lifespan, right? Because to live long, you got to be protected from cardiovascular disease. You got to be protected from cancer. You got to be protected from neurodegenerative disease. You got to be protected from risky behavior. I mean, it's so multifactorial, it's unbelievable. So I don't think big population studies are actually going to turn up things that are very predictive. I think the fact that I think you're going to find things that contribute or prolong lifespan in smaller groups, private mutations that exist in families, and this Amish kindred is a big, large, extended family where everybody's at least remotely related. Yeah.
Philosophically, do you think we miss the opportunity to get people to lower their blood sugar and insulin, which would help all those aging markers by focusing on this drug as the answer to anti-aging? And do we encourage people to look for a drug instead of focusing on lifestyle?
Yeah, okay. That's perfectly appropriate question. The question is, do we do everybody a disservice by focusing on a drug that might prolong your lifespan rather than taking care of yourself, keeping your weight under control, doing other things? Of course, that's reasonable. And I have no desire to take this drug and test it in healthy people and say, can we prolong lifespan? I do want to take it into those people that I see every day in the clinic that have accelerated aging, people with chronic kidney disease, that age right in front of my eyes, that have heart attacks at 25 and 30, and nothing you can do with their diet or their lifestyle is going to change that. So we want to go impact on diseases that look sort of like accelerated aging, but not aging itself. I'll leave that to somebody else to do it. In fact, that'll be beyond my lifespan. Yeah.
Are there things available that will increase the inflammation response?
Are there drugs available that will decrease the inflammatory response? Well, of course there are. There are simple drugs that you can buy over the counter. They're anti-inflammatories, non-steroidal anti-inflammatory drugs will reduce the inflammatory response. There are very high-tech anti-inflammatory drugs like etanercept, infliximab, drugs that block TNF-alpha. They're very, very potent anti-inflammatory drugs. There's also a risk there because you get some risk of infection when you take those things, particularly infections with things like TB. So you got to watch very carefully. But we give those drugs all the time for rheumatoid arthritis and Crohn's disease and things like that. Now, maybe do we need to wrap up? Or we okay? One more? One more question? Back here?
Question. Do you think the effect of iron deficiency on aging must be related to thrombosis prevention? And therefore, do we know, for example, the patients with non-life-threatening disease, like Hemophilia or do they have longer life expectancy?
The question is, do I think that the effect of PI1 deficiency on lifespan has to do with its antithrombotic effect? No, I don't. I actually think it has to do with its insulin effect. I think your PI1 level determines your insulin level, and that's what the Amish tell us. So their insulin levels are significantly lower for the same BMI. That tells me that although PI1 tracks with insulin, and people have always said, well, insulin drives PI1, actually the Amish tell it's the opposite. If your PI1 level is low, your insulin level is going to be low, and I think that's what contributes to their protection from aging. But, I could be wrong. Thanks a lot again, everybody.